Hello and welcome to this webinar titled Preview of USP Informational Chapter 1085, Guidelines on the Endotoxin Test, hosted by Biopharma Asia magazine and presented by Karen Zink McCullough, Principal Consultant at MMI Associates, and Kevin L. Williams, Senior Scientist Endotoxins at bioMérieux USA. My name is Orshaya Balog and I will be the moderator. Now please allow me to introduce our first presenter, Karen Zink McCullough, Principal Consultant at MMI Associates. Karen Zink McCullough is the Principal Consultant at MMI Associates, a firm focusing on GMP and microbiology in the drug, biologics, cell therapy, and medical device industries, with a specialty in the bacterial endotoxins test. Ms. McCullough was the first to publish on the use of the BET as a replacement for the rabbit pyrogen test for small volume parenterals. She has published over 50 articles and book chapters, and she is the editor of the book The Bacterial Endotoxins Test, a Practical Guide. She is a well-respected international speaker on GMP and the BET. Ms. McCullough is a chairperson of the LAL Users Group, is a United States representative to work in Group 2 TC 209 for the revision of ISO 14698, and is a member of the United States Pharmacopoeia Expert Committee on Microbiological General Chapters. She holds a bachelor's degree in bacteriology from Rutgers University and a master's degree in molecular biology from the University of Oregon. Welcome, Karen.
Thank you very much, Ursula, and thank you to bioMérieux and to Biopharma Asia for this opportunity to chat with you today. I'm going to be talking a little bit about the USP's informational chapter. We like to call it Chapter 1085 because it matches up with Chapter 85, which is the bacterial endotoxins test. So before we get started, I'd like to make a disclaimer here that while I was on the committee that drafted this informational chapter, any opinions that are expressed here by myself is just my opinion and nobody else on the Microbiology General Chapter's Expert Committee. I also want you to know that this informational chapter is going to be published in the July and August issue of Pharmacopeial Forum. And the reason why we're doing this webinar today is to put out there in the public what we're doing here so that everybody knows and hopefully will be able to respond when this is published. When it's published in Pharmacopoeia Forum, we will be asking for public comment on the chapter. So why are we doing this informational chapter? Well, if you remember, if you're old enough to remember, there was a guideline published in 1987 by FDA, and it was retired in 2012, and it was replaced with a series of questions and answers. And upon the retirement of that 1987 guideline, we felt that there were some gaps in the written literature about how to execute an LAL test. For example, there's nothing in the literature that tells us how to do an RSE-CSE standardization. There's confusion regarding calculating endotoxin limits. The 87 guideline had some suggestions about training in it, but that was lost with the publication of the 2012 guidance. And the 1987 had its own spin on resolving OOS results, but we take a different look at it in the 1085 chapter, and we try and relate it back to FDA's 2006 guidance on OOSs. In addition, we get a lot of questions at USP from users of Chapter 85. And as a consultant, I get a lot of questions, and we recognize that formulations have changed, and so we think it's time perhaps for some sort of an informational chapter. So let's talk about the RSE standardization, RSE-CSE. The current primary and secondary standards are made from purified lipopolysaccharides. And those purified lipopolysaccharides are extracted and purified from the cell walls of gram-negative bacteria. In the case of the RSE, it's E. coli 0113. In the case of some of the control standard endotoxins, the secondary standards, it's a different strain of E. coli called 055B5. Now, the RSE, which is the reference standard endotoxin, is the primary standard. and that standard now is consistent in the European Pharmacopoeia, the United States Pharmacopoeia, the Japanese Pharmacopoeia, and the World Health Organization. That wasn't always the case, but it is now. And so one endotoxin unit, as we tend to call it in the United States, is equivalent to one international unit, which is what they're called in Europe. The control standard endotoxin, also called CSE, is the secondary standard. Now, generally, the secondary standard is provided by lysate manufacturers. So if you buy a kit from a lysate manufacturer, you will have your lysate reagent, you'll have some endotoxin standard. The lysate manufacturer purchases that lipopolysaccharide from a third party, and then in some cases, or to different degrees, they bring it back and they further formulate it into a control standard endotoxin that you find in your kit. Just so that we understand, the lysate is regulated by the Food and Drug Administration here in the United States, but endotoxin standards are not. The Redforth endotoxin standard in the U.S. is held by the United States Pharmacopoeia, but it is not regulated by the Food and Drug Administration. So what is the standardization? Well, RSE being the primary standard is filled in endotoxin units per vial. If you were to order some of the standard, you'd see that it would say there's 10,000 endotoxin units per vial. CSE is a secondary standard, but it's not filled in endotoxin units per vial, it's filled in nanograms per vial. So what this standardization does is it normalizes or compares the activity of the CSE in nanograms per milliliter to the activity of the RSE in EUs per milliliter to result in a potency. The potency is expressed in EUs per nanogram. Now, the lysate manufacturers generally do this for you, so you will get a certificate of analysis in your kit to say that with this particular lot of lysate and this particular lot of endotoxin, the potency of the endotoxin is so many EUs per nanogram, or they will tell you to reconstitute your vial of CSE with so many milliliters of water to result in a concentration of 50 EUs per milliliter or some such instruction. Got to remember that the potency of the CSE is dependent on the lysate lot that you use. So if you change one or the other, if you change the lysate lot or if you change the CSE lot, the standardization needs to be done all over again. And that's a little bit of a tricky thing to think about, but it's true. It's all dependent on the lots of the unique combination of lots. So how do we do this for gel clot? Well, we're going to dilute the RSE in EUs per milliliter to bracket the label claim sensitivity of the lysate. We're going to dilute the CSE in nanograms per milliliter so that we get an endpoint. Now, what's the endpoint? An endpoint is, in any dilution series, the endpoint is the last positive tube in the series that's followed by a negative. It must be followed by a negative in order to be the endpoint. So what we'll do is we'll dilute the CSE in nanograms per milliliter so that we get an endpoint. And we're going to assume that there's between 5 and 20 EUs per nanogram in the CSE. What we're going to do then is calculate the geometric means of the two test results, and we're going to compare those. So what's the geometric mean? If I take the endpoint in EUs per milliliter, and I convert those to log 10, then I can calculate the mean of the log 10 values and take the anti-log of that, and that is the geometric mean. So for example here, if I have a lysate with a lambda, an endpoint of 0.125 EUs per milliliter sensitivity, and I take and I do my RSE on the left-hand side diluted in EUs per milliliter, those red crosses are the endpoints. So for replicate 1, the endpoint is 0.125 because it's followed by a negative at 0.06. Replicate 2, the same. Replicate 3, the endpoint is 0.25 because it has a negative at 0.125. Replicate 4 is the same. Over on the other side, there's the CSE diluted in nanograms per milliliter, and you can see that there's different endpoints here as well. The first one was at 0.006 nanograms per milliliter. The other three were at 0.0125 nanograms per milliliter. So if I take the RSE, the geometric mean of the RSE, and divide it by the geometric mean of the CSE, I end up with 18 EUs per nanogram. And that is the potency of the secondary standard with that particular lot of lysate. And again, if the lysate lot changes or if the endotoxin lot changes, we have to redo all of this. What if I have a kinetic test? So you can see there on the left that we have a sort of a generic looking standard curve. And what we do then is we take our CSE and we dilute the CSE in nanograms per milliliter, and we treat those as unknowns. So what we can do then is find a dilution of the CSE that falls on our standard curve, okay, and then it will give us a value in EUs per milliliter. Remember, all endotoxin stuff, all BET test results are in EUs per milliliter. But we need to change that to EUs per nanogram. So what do? We say, okay, here's our interpolated value in EUs per milliliter, and here's the corresponding known value in nanograms per milliliter of that same dilution. And then we come up with EUs per nanogram. And again, this becomes lysate lot dependent. So every time we change either the CSE or the lysate lot, we have to do the same thing. In this case, the reference standard endotoxin is used to make that standard curve. And that's how we compare the RSE with the CSE, because the standard curve is made with the RSE. And then we're looking to find the potency of our CSE. What about if I have a liquid preparation? If I have a liquid preparation, we don't know anything about weight. So the liquid preparation doesn't have a potency. The liquid preparation has an activity. It's a concentration of activity of EUs per milliliter. So in that case, if I had that, I would just test my liquid preparation as an unknown and find out how many EUs per milliliter I have, because I'm not going to know how many EUs per nanogram I have because I don't know the weight of the stuff that's in the liquid preparation. So liquid preparations don't have a potency. They have a concentration. The concentration is expressed as EUs per milliliter, so it's an activity-based value. We get a lot of questions about calculating endotoxin limits, and this gets really complicated. If you look at many package inserts, there are various different ways in order to administer some products. And when you calculate the endotoxin limit for your product, you have to always go back to the package insert, look at the various different ways that it can be administered, and then calculate the endotoxin limit for each of those ways that it can be administered. And then you pick the most stringent one, and that becomes the endotoxin limit for your product. So let's talk a little bit about endotoxin limits. There are going to be examples in 1085 of calculating endotoxin limits for drugs and biologics and medical devices. Medical devices are in EUs per milliliter, and that's based on the elution volume and the test method sensitivity. There's going to be some endotoxin limits proposed for combination products in this new chapter. And large volume parenterals are generally 0.5 EUs per milliliter, with the exception being water for injection, which the compendial limit for that is 0.25 EUs per milliliter. So let's look at the endotoxin limit calculation. It's usually K over M is the formula, where K is the threshold pyrogenic dose. What's that? That's the amount of endotoxin in terms of activity it takes in order to induce a fever in a rabbit. Now, that's not the direct measurement. Actually, it's a statistical measurement. It's really I think it's the lower 99th percentile of the mean pyrogenic dose when this was done years and years ago with lots and lots and lots of rabbits. But in any case, it's a very conservative value that's been used called the threshold pyrogenic dose, and it's been used for many years. So the threshold pyrogenic dose is 5 endotoxin units per kilo per hour. And again, that was derived from a lot of rabbit testing that was done years ago and pretty much confirmed in humans a bit more recently. M is the maximum human dose in units per kilo per hour. If the maximum human dose is a pediatric dose, then we work with the pediatric dose, whichever one is more stringent, the adult dose or the pediatric dose. So here's a table, and we're not going to go through all of this stuff on the table, but basically it says for various routes of administration, what is K and what is M, so that when you go to calculate K and M, you know exactly what the values are. It's important that you keep all of the units here, because as you go through this and you start to cross out the units, you'll know in the end if you got the right answer, because the units will match. If you don't pay attention to the units, then you might end up with the wrong answer. So while we're not going to go through this, I ask that you take some time and take a look at this. So here's a question. We have a product and our dose is 10 milligrams. What's the endotoxin limit? Well, next question is, are we missing something? And the answer to that is yes. We're missing the particulars of the dose. Now, the dose, remember, we have to worry about the route of administration, because the endotoxin limit changes with the route of administration. For intramuscular and intravenous injections, it's 5 EUs per kilo. For intrathecal injections, it's 0.2 EUs per kilo per hour. So we have to worry about the route of administration, and we have to worry about the time of administration, because we're looking at the amount of endotoxin that can be introduced into the person in the course of only 1 hour. So let's take a look at some things. If I have a dose of 10 milligrams per kilo per hour administered intramuscularly as a bolus meaning all at one time, so within the hour, K over M is 5 EUs per kilo per hour divided by 10 milligrams per kilo per hour, and then we end up with 0.5 EUs per milligram. So that one's pretty easy. Supposing we have a dose of 10 milligrams per person administered IV as a bolus, meaning within 1 hour. Well, remember, we always have to reduce everything back to EUs per kilo per hour, and our dose here is milligrams per person. So what do we have to do? Well, we have to say, okay, let's assume that the average adult in the United States weighs 70 kilos. Now remember, other populations may have different weights. So in Japan, for example, the average person weighs 60 kilos. If you have a product that's targeted to children, you might want to go to the CDC tables to find out how much the average child weighs. So if your drug is targeted to adolescent children, you can go to those tables and find out what the average weight of the adolescent child is. But in any case, when you have a dose that's in milligrams or milliliters, whatever, per person, we have to correct that dose to per kilo. So in this case, we take 10 milligrams per person, we divide it by 70 kilos per person, and we end up with 0.14 milligrams per kilo. Now we can use that as the denominator in our endotoxin limit calculation. So K over M is 5 EUs per kilo per hour divided by 0.14 milligrams per kilo per hour, and this endotoxin limit is 35.7 EUs per milligram. Here's another one. The dose 10 milligrams per kilo administered IV in 4 hours. Well, remember, all of the endotoxin limits are EUs per kilo per hour. So now what we need to do is say, okay, so this is what we can deliver in 4 hours. How much can we deliver in 1 hour? So we have to correct the dose to say if I can get 10 milliliters per kilo in the space of 4 hours, that means I can have 2.5 milligrams per kilo per hour. And then I can plug that into my K over M, and the resulting answer is 2 EUs per milligram. So you can see where we're going here. We're not going to go through all of these because there's not time, but you can see where we're going. We have to look in the package insert and understand very carefully how this drug product is being used. And then we have to calculate endotoxin limits for all of the ways that it can be used, and then choose the most stringent one as the endotoxin limit for the product. Here we have Here we have a dose of 10 milligrams per kilo administered intrathecally. Remember, K for intrathecal is 0.2 EUs per kilo per hour. So in this case, we're allowed 0.02 EUs per milligram. Here's a dose administered 10 milligrams per person administered as 4 hours. Whoa. Now we have to correct for two things. We have to correct for per person back down to per kilo, and we have to correct for 4 hours back down to 1. So we end up that. In the end, you can see here all of these various ways that we can think about the endotoxin limit for a dose of 10 milligrams. We end up with a lot of different endotoxin limits: 0.5, 35.7, 2, 0.02, and 142. So we have to really pay very much attention to the dosing. There is a guidance out there for a cellular therapy guidance that's out there right now, and it talks about the endotoxin limit as being 5 EUs per kilo. But that's not the whole thing. We have to understand the dose. So remember that 5 EUs per kilo is not the endotoxin limit. The endotoxin limit is dose dependent. So this is part of what's going to be in the informational chapter. Back when the 1987 guidance was provided, there was suggested training. Okay, now that's gone with the 2012 question and answer. So we're going to make some recommendations. We think that there should be an understanding of the test, meaning that there should be a classroom training, a seminar, something to read, something to absorb, something to understand a little bit about the test. For example, we know that there's a bunch of things that can interfere with the test, one of the big ones being pH. So if the analyst doesn't understand that pH can affect the results of the test, then he or she, without knowing anything different, might go through and get a false result because the pH is off. So we want the training to include some classroom training in addition to proficiency training. Proficiency training meaning to confirm the label claim for gel clot or to prepare a linear standard curve for kinetic tests. We also put in there a provision for retraining. When should people be retrained? Well, we think that failure to meet the requirements of the initial training is one time. Let's say we have a number of invalid tests, or we have a number of OOSs, or we have a bunch of erratic results. If you have a quantitative test and you see erratic results in terms of the slope and the y-intercept, well, then perhaps that person, that analyst needs to be retrained because perhaps that analyst doesn't understand how to make dilutions correctly, or perhaps that person doesn't understand how to work the pipetter correctly, or something like that. So whenever there's erratic results, whenever there's OOSs, whenever there's a bunch of invalid tests, you want to look back and ask, does this analyst need to be retrained? All right, so let's talk a little bit about OOS. So in the 1987 guidance, it said, well, if you have a failure, it said, oh, okay, that's okay, go back and test 10 units individually. If all 10 pass, then you're good to go. And that worked for 1987, but that doesn't work for 2018, right? That could be called testing into compliance these days. So what are we thinking now? So consistent with the 2006 guidance, we think, okay, if there's a phase 1, we should first look at the laboratory and say, do we believe that the laboratory data are accurate? So then there's a whole checklist that you can go through, and this is all explained in this chapter, whole checklist that you can go through. What would you check in order to see if the data in fact are accurate or if there could have been some error in generating those data? If you find that those data are correct, there's no reason to invalidate those data, then we suggest that we follow the guidance again and we look to phase 2 and expand the investigation to include manufacturing. So just like any other OOS, first take a look at the lab, make sure the data are accurate, and then you can go forward and test some hypotheses and expand the investigation. In the guidance document, it says that this footnote, there's a footnote in the guidance document that says that the principles in the guidance aren't applicable to biological tests. And I think the details in the guidance are not applicable to a biological test, but I think that the principles are, meaning that we can't test things into compliance. And even though USP says to average test results for photometric assays, I think we need to be careful that we don't average into compliance as well. So for example, if my limit is 1 EU per milliliter and I'm doing a kinetic chromogenic test, and my 3 test results are 0.87 EUs per milliliter, 1.02, and 1.1, the average is 0.99 EUs per milliliter, and theoretically we pass. But in the LAL world, 0.87, 1.02, and 1.1 are not different numbers. So in the best case, all of those 3 values suggest that the result is very, very close to the limit. And we need to understand that more significant figures in the data reported do not make the test result more significant. So significant figures really becomes important, but in this case, the more significant figures doesn't increase the accuracy or the precision of the test. So in our case here, we had 2 out of 3 that were above the limit and 1 that was below the limit. Regardless of where that average is, we have to look at those 3 values and say that we're very, very close to the edge. And so should this product pass or should this product not pass? We talk a little bit about outliers. Some firms say, oh, we're going to test in triplicate so that we can throw one out if it doesn't match the other 2. Well, actually, that's really not the reason that you want to put in a third replicate. The third replicate incrementally makes your average a little bit more accurate or precise, but it's not there so that you can throw one out. So be careful, because you're going to need a good reason to invalidate a test result. And if you say because it doesn't match or because it adversely affects our average, that's just not a good reason. So we talk about that in the chapter as well. So if we have an OOS, there's a bunch of questions that we might ask. Has this happened before? And just like any other kind of an investigation, the pattern or the trend is much, much more accurate than any given test result. Remember, LAL tests are not hugely accurate. There's a significant wiggle room in those test results. So remember that, and so you want to say, okay, the trend becomes much more important than any one individual test result. So have we seen this before? If so, we start to ask, well, do we have a new manufacturer of materials? Do we have new suppliers of raw materials? Do we have new operators? Is there a new analyst? Is there new reagent lots, new test methods? In other words, we're going to ask what's different about this test result than for the previous test results. And that's where we're going to look to find out what went wrong. We suggest in the chapter as well that you use a graphic organizer when you do an OOS investigation, because when you use a graphic organizer, such as a fault tree, do a fault tree analysis or a fishbone analysis, you can brainstorm ahead of time with a cross-functional group of people so that we can think about where all of the possible problems could occur, and then we can put them down and then just go tick them off as we do the investigation. So we recommend the use of some sort of a graphic organizer. We recognize as well that you're not always going to find a root cause. In fact, probably more often than not, you're not going to find a real smoking gun root cause. But just because you don't find a root cause, it doesn't relieve you of the obligation to look for one. So every OOS result has to be taken very seriously. So this chapter is going to be published in the July and August edition of the Pharmacopoeia Forum, and the Pharmacopoeia Forum is free. So if you go to the USP website and sign up, you can get access to Pharmacopoeia Forum for free. And this chapter aside, there's a lot of valuable information in the Pharmacopoeia Forum. So we're asking that you sign up. You please read and comment on our proposed chapter. One slide that I think I skipped inadvertently is chapters that are numbered below 1,000 are called enforceable chapters. So chapter 85, the USP bacterial endotoxins test, is an enforceable chapter. Chapter 161, which is the medical devices, pyrogen and endotoxin testing, that's an enforceable chapter. Chapter 1085, it's over 1,000. So this chapter is not enforceable. This chapter is there only for information. Just like there's a whole series of depyrogenation chapters called 1228.x, X being the number of the chapter for different kinds of depyrogenation methods. Those are for information as well, but they are not enforceable. So I urge you to sign up for Pharmacopoeia Forum and to take advantage of the low cost, meaning the free, of the PF. And then when this does come out in July and August, please take some time to read it and give us some feedback on it. We much appreciate that. So I'm going to say thank you, and I believe we're going to hold the questions until after Kevin's presentation.
Thank you, Karen. Once again, I would like to remind our viewers that there will be a live question and answer session at the end of the presentation. Please feel free to send in your questions at any time during the presentation via the Questions tab located at the right-hand side of your screen, and we will go through these at the end of the presentation. Now, please allow me to introduce our second presenter, Kevin L. Williams, Senior Scientist Endotoxin at bioMérieux, USA. Kevin studied microbiology at the Texas A&M University and has worked in the pharmaceutical field for 35 years. The bulk of his career was spent at Eli Lilly and Company developing QC tests for microbial and endotoxin detection. Kevin's current role at bioMérieux as senior scientist focuses on endotoxin science and method development, and he has also helped setting up a dedicated endotoxin service laboratory in the USA. He is the author of Endotoxins 2nd edition and Endotoxins 3rd edition. Welcome, Kevin.
Thank you, and thank you for calling in today. I want to talk about regulatory compliance of alternative methods, but I'm going to start out with a little bit of background information on why it might seem natural for recombinant factor C to be the next test. Look at the mechanism of action. Then the bulk of the talk will be on regulatory expectations for alternative validation, since it's not a USP BET 85 compendial method yet. And then also talk a little bit about a time, labor, and error saving product that is that uses RFC that bioMérieux has just come out with. So if we're wondering why recombinant factor C might be the next logical step in endotoxin testing, we can see that as a medical paradigm, the replacement of natural proteins with recombinant proteins has been ongoing for some time. In 1982, Lilly's came out with recombinant human insulin, so that replaced the need to harvest insulin from pigs and cows. And I saw a paper that described that it took 2 tons of pig parts, as they called it, to make only 8 ounces of insulin. So you can see that's a laborious and not very efficient process. And I believe eventually the RFC will be the way to go in terms of recombinantly producing that material. It's the basis of the biotechnology revolution that's been ongoing for some time. There are monoclonals, cytokines, blood factors, enzymes, all kinds of drug products that are produced by recombinant technology. So it makes sense that the LAL factor C would be cloned and produced that way. So it's also RFC is not only conservation but also a modernization prospect. If we look at the cascade of the reaction that comes from the horseshoe crab, that is the means of detecting endotoxin and LAL on the left-hand side, we can see there are a number of proteins in there. There's factor C, factor B, factor G, the so-called beta-glucan detecting biosensor. There's proclotting enzyme, coagulagen. So there's a number of proteins there. And with the recombinant factor C, only the factor C has been cloned from the horseshoe crab and produced synthetically. So it's the only biosensor present in the LAL. Let's see. The activated recombinant factor C cleaves a fluorogenic substrate directly instead of relying on factor B, which is in the horseshoe crab cascade. So let's look a little bit more detailed to that reaction. Just one slide on the mechanism. So RFC is what they call a zymogen, so that it exists in the horseshoe crab hemolymph as a very large protein, 123,000 Daltons. You might compare that to human insulin, which is 5,800 Daltons. If there's no endotoxin present, then this is what the protein looks like. It's got a lot of different domains to the protein. When we say that when endotoxin binds the molecule, the protein, the factor C molecule, it binds at a certain specificity in the sushi proteins, it's been shown. So this activates the zymogen to its enzymatic form. It breaks some bonds. It rearranges the serine protease on the other end of the molecule, and this chymotrypsin-like activity then exposes an active site. Trying to click the button here. Exposes a serine protease active site, which makes the active site available to the next factor in the cascade, which here is factor B. And the only difference being in the RFC test, it releases the fluorophore instead of activating another cascade part. We got some feedback from FDA in 2012 with the so-called replacement of the long-standing 1987 guide that Karen talked about with the so-called Q&A guideline, and they dedicated a couple of questions to alternative validation. So FDA says that they are looking for the user to understand the advantages that they are presenting to be proposed with the new method, and also they're saying to validate using both USP 1225 and also USP 85. USP 85 because it's such an analogous test. They do remind the users that the gel clot test is the compendial assay, but they also state that they're not looking for a comparison to the gel clot test. If you remember many years back, the kinetic or photometric tests were also alternative tests, and we validated those before they were USP tests, and then when they converted to USP tests, we had already been using them for some time. In the vernacular of the compendia, the normal BET85 is a verification because you're not really validating the assay, you're just verifying it under the conditions of use. So the validation of the compendial procedures, users are a little less familiar with. So we'll go through 1225 a little bit. But the most basic question to be answered of alternative validation is just, does the new test provide the same results, approximately the same results as the old test? So can it serve in lieu of the old test? A couple of caveats here. If we're just comparing the new to the old, we're not really encompassing all the variability. We would just be comparing two supplier tests. But the question remains, are the expectations of the compendial test being met? So if you have a method, or if you don't have a method, or if your current method is gel clot, you don't have to set up a method. You establish the new method according to the expectations of the BET compendial test. So that's a little confusing for users that they think they have to test side by side. Although we're going to see something that looks like a contradiction, but it's not quite here in a minute. So FDA is wanting to look at the limit of quantitation, because it's not a limit test, and also look at the inhibition enhancement as per BET 85. These things are also gone over in the ICH harmonized guideline on validation. So between the two, you can get a better idea of what they're expecting. So they say, in addition to using spike samples, a battery of field samples of product found to be positive may be a good source for comparing results. So if you have endogenous activity, then you would want to compare in a so-called side-by-side test, because the testing at the specification is very important. So you would want to know the answer to that. But most often these days, we don't see a lot of activity in our testing, especially of finished products. So therefore, you would not need to do that. The FDA says that they're looking for advantages, so the user should be aware of what the advantages are. And there are many advantages we can go through. The specificity, if beta-glucans are a problem, we saw in the previous slide how RSC takes away the factor G, so you can't detect glucans in these tests. So specificity is more specific for RSC. We have high standard curve linearity and curve-to-curve reproducibility. So if you put these standard curves together, they really sit right on top of each other without much natural variability. We're going to talk about in the next few slides some product-specific advantages because product to product, you can see some tests works better than others, even within LAL itself. We're going to talk about sustainability and conservation. Now, this is a real-world consideration and not really a USP parameter, but it's important. It's an important consideration, as we saw in that modernization paradigm. Also, in terms of automation, the RFC mix is extremely stable for several hours. This is something that you couldn't do with LAL, is leave it out on the bench while the robot goes through its tasks over time. And also, as an endpoint test, you can set up testing to do a first read, put that read in an external incubator, perform a second read. In this way, you can do more of a high throughput testing, even though the assay may take an hour. You're not waiting for it to start the next assay. So there's many advantages that can be seen. And when we talk about variability, of the compendial method, we know LAL has its own variability. So this testing is not expected to be perfect, just comparable. Okay, in terms of the equivalence that needs to be demonstrated, USP 1225 gives these so-called data elements. So accuracy just means that it's close to the truth, and the truth here is the reference standard. So we would dilute down different concentrations of the RSE and demonstrate that it is accurate. Precision, as shown in the little target at the bottom, precision is how close the values are together to each other. So you could have a test that's precise but not very accurate. So they're looking for accuracy and precision. Specificity, we've already talked about that. Measures what it's supposed to. Since it's a quantitative test, we don't look at the detection limit. We look at the quantitation limit, or the limit of quantitation. This should be accurate and precise around the sensitivity, also called lambda. The linearity, that's something everyone in BET testing is familiar with, and range as well. That's just the upper and lower levels of the analyte that can be detected. So there's more than one way to understand this testing. My favorite way is to look at it as per the older IQ, OQ, PQ performance. So we can see that on the left-hand side, we have some questions to answer. We'll look at the supplier method, understand the instrumentation. So just really familiarization of the user with the testing. And then we'll want to set up the reader, validate the equipment, and install the software. So all the things that you would do to set up a BET test is very analogous here to set up for RSC testing. Really the only significant difference is the use of a fluorescent reader instead of an absorbance reader. PQ1 is what here we're calling the method validation. So the method validation would use non-interfering water to demonstrate these accuracy, precision, limit of quantitation. These 1225 data elements can be proven. We call this the method validation. So once the method validation is done, it won't have to be revisited. But what we go to is a product-specific validation, and this also is very analogous to BET because we would look at the non-interfering concentration test, and then we would perform the 3-lot validation. So most users are familiar with this methodology. Just to reiterate, some of these data elements can be referenced either from other studies or from the supplier validation, and I would recommend ruggedness and specificity be among those. Unless you have specific things that you want to show, then it would be good to use those. The PQ1, it's an abbreviated study to verify that the expected results are occurring. So this is the method validation. This is a little repetitive, but it's good to go through it again. And then PQ2 is your product-specific validation, very analogous. So the PQ2s you would validate once you had your method done for any new product or new excipient or new water. You could perform that without having to go back and worry about the USP 1225 validation, method validation. The timeline, people generally think this takes longer than it does. If you do your homework and get the testing organized, you can really get it done in 3 half days of testing. So if we look at the PQ1, the USP 1225, accuracy, precision, linearity, these can be evaluated by a single setup repeated 4 times. 2 analysts, 2 days, and 2 reagent lots are cross-varied, and we'll show the template setup for that. The specificity, we already talked about that. It's good to show if you have a concern with beta-glucans. You can certainly show the demonstration of that. Otherwise, I would reference it from the supplier validation. The same with the ruggedness. And the product-specific validation, the LOQ is lambda. It can be performed at 005, but also it can be performed at 05. Say for water testing with a higher limit, you can have a 20-minute test. Or if you have some interference that needs to be overcome, you can certainly lower the lambda to 0.001. All these have been validated and can be validated for your testing too. The NIC test, typically 2-folds or 10-folds to show where the interference is overcome, and this is what BET 85 recommends for LAL testing, so we've taken that to apply it here. And also the IE testing, which is your verification testing very analogous. So here's a look at the software template setup. You can see how we've put a couple of standard curves on the same plate. Here we have a 60-minute test that goes down to 0.005. Let's say you want to validate that along with the 001 test to have some flexibility in your choices. You can certainly do that, or you can choose the faster 20-minute test, which goes down to 0.05. So you validate simply what you're going to be using, validate your method, and then you've got 3 different non-interfering waters on here. These values, such as 0.0015, of course, you would use the data for the ones that correspond with the lambda that's appropriate. You couldn't expect to get 0.0015 on an 0.05 curve. So this is putting it all together to get an efficient validation. bioMérieux provides validation protocols and formulas to capture the user results. So it really is a complete package to perform what is otherwise can be a confusing exercise. Here's a very recent test out of Japan on the RFC specificity. And the English is down there. Some of you may be able to read the Japanese, but not me. A collaborative study on the BET using recombinant factor C procedure for LPS detection. So let's look at that. Table 2 really shows the meat of the demonstration, and they use naturally occurring, only slightly purified isolates, 1 through 5, as you can see, a range of organisms, and they tested them by 3 LAL methods and 3 RSC methods. In 6 through 11, you can see the same testing performed on some natural waters, and we have these translated here on the next slide. So the natural waters, lake water, river waters, wastewater, mineral water, and just a water. Not sure what that one is. But I put these numbers into Excel, and you can see there's not 2 different sets of numbers. There's not a set for LAL and a set for RFC. These are thoroughly mixed together in an equivalence demonstration, so you can really see the demonstration of equivalence of the recovery here. In terms of a new product to make testing easier, I call it semi-automated test, where the standard curve is already pre-dried into the wells, and also the positive product controls are dried into the wells also. So really you just have to add water to the standard curve and samples to the sample plates. And you also have the choice here to use the standard curve that best suits your product. So this one runs from 0.5 to 50, but you can also choose 0.5 to 5, or what's most commonly used, the 0.05 to 5. So you can see some flexibility built into the plate, and then all the sample PPCs are performed at 0.5. If we look at the ease of use of the plate, we can see the steps we're leaving out. The first step, adding water and samples to the dedicated wells. So we're leaving out here the reconstitution and the vortexing of the CSE. That's 10 minutes. The preparation of the standard curve, another 10 minutes. And also the addition of the PPCs. So these are, to me, the most error-prone parts of the test. So you come with the test that you can expect to be perfect every time, and there's been a lot of validation that's shown I can't say that I've even seen a R value of less than 1.000 yet. So these are very promising, and bioMérieux is very proud to come out with this product. And also, since the BET85 is part of the alternative validation, I wanted to show this inhibition enhancement recovery values using different kinds of products. Here are a bunch of excipients that we're tested at 2 different concentrations. So you can see sodium chloride, for example, tested at a more concentrated and less concentrated. The plate did a good job of recovering it. Tween, also the beta-glucans, easy to demonstrate the trouble that it causes with LAL, and the fact that the RFC is ignoring the presence of the glucans. Drug products, here's a specific monoclonal and a human serum albumin, which is a addition to some drug products. So, it's gone pretty quickly. To summarize, the natural to recombinant protein transition is an established medical modernization paradigm. So that really answers the why RSC may be the next logical step in endotoxin testing. We can see the how is answered by a simplified test in which it's a direct test of the detection by the biosensor. We've gone over the timeline. Really, it's just 3 half days of testing once you've organized that testing with the bioMérieux templates and software templates and data capture forms. And lastly, we have some help here. Once you've used the endozyme to go, you feel kind of spoiled and you don't want to go back to making curves and PPCs. So that's a good addition to the market. And then bioMérieux supplies the readers, the training, the SOPs, and the alternate validation documentation materials. So I think that's the last slide. So I thank you for joining today.
Thank you, Kevin. Now we will begin the question and answer segment of the webinar. Once again, I would like to remind the audience that you can still send your questions via the questions tab on the right-hand side of your screen. Our first question is, Karen's company makes a combination product. Does this chapter provide guidance on limits for combination products? Karen?
Yes, it does. It speaks to all kinds of different combination products and proposes limits for these combination products, yes.
Thank you. The next question is product side-by-side testing required? Kevin?
It's not required, especially if you don't have endogenous endotoxin in your samples. But if you do have endogenous endotoxin, then it's recommended because you really just want to know that your specification is how your test reacts relative to the specification.
Thank you. Will 1085 have guidance for overcoming test interference? Karen?
Yeah, it does. It has a section in there on overcoming basic interferences. You know, like I said in the discussion, pH is a basic interference, and so 1085 goes into some of the basic interferences and how you might be able to mitigate some of those basic interferences. Yep.
Thank you. Should you use CSE or RSE for the compendial method if you perform such a side-by-side test? Kevin?
For the side-by-side testing, you're really wanting to see how close the values are for the new and the old tests. So I would use RSE for both of those tests to take out the little bit of difference from the CSE.
Thank you, Kevin. Before I finish the webinar, I would like to ask our presenters, Karen Zink McCullough and Kevin L. Williams, if you have any closing remarks. Karen?
No, I just want to thank everybody for attending today. Thank Biopharma Asia for this opportunity, and thank Kevin and bioMérieux for asking me to help today as well. And like I say, just keep your eye out for the 1085 chapter that's going to be in Pharmacopoeia Forum, and please take the time to comment and send us some feedback.
Thank you, Karen. Kevin, do you have any closing remarks?
Just to thank you to Karen for performing that USP, I guess call it an add back of the old 1985 important parameters. And then thank you to Biopharma Asia for the hosting the event, and also thank everybody for calling in.
Thank you, Kevin. I would like to take this opportunity to thank our presenters, Karen Zink McCullough and Kevin L. Williams, for sharing their knowledge with us. I would like to remind our audience that you can view this webinar on demand by visiting biopharma-asia.com. Thank you, everyone. Have a great day.